Welcome to my channel XY Biology Academy. Now that we've understood how a bright field microscope forms an image, let's move on to another important type of light microscope, the dark field microscope. Although both microscopes contain almost the same optical components, they produce completely different images. Why? The answer lies in how light is directed toward the specimen. Before we follow the ray diagram, let's first understand why it is called a dark field microscope. In this microscope, the background, or field of view, appears completely dark, while the specimen appears bright. This happens because only the light reflected and scattered by the specimen reaches the objective lens, whereas the direct transmitted light is prevented from entering it. That is why it is called a dark field microscope. Now let's compare the ray diagrams of the two microscopes. In a bright field microscope, the condenser produces a solid cone of light that passes directly through the specimen. The transmitted light enters the objective lens and forms the image. In contrast, the dark field microscope uses a special condenser that blocks the central rays and produces a hollow cone of light. As a result, the direct transmitted light misses the objective lens completely. Only the light reflected, refracted, and scattered by the specimen enters the objective to form the image. Now let's follow the complete ray diagram of the dark field microscope. The process begins with the light source, usually an LED or tungsten lamp, located at the base of the microscope. The light travels upward and enters the dark field condenser. Unlike a bright field condenser, the dark field condenser contains an opaque circular disc, also known as the dark field stop, positioned at its center. This opaque disc blocks the central rays of light and allows only the peripheral rays to pass. The condenser then refracts these peripheral rays, producing a hollow cone of highly oblique light that is focused toward the specimen. Now imagine that no specimen is present on the slide. Since the light is traveling at a highly oblique angle, the rays bypass the objective lens without entering it. As a result, no light reaches the objective, no image is formed, and the entire field of view remains completely dark. Now place a specimen on the slide. When the hollow cone of light strikes the specimen, part of the light is reflected, refracted, and scattered in different directions. Some of these redirected rays now enter the objective lens. The objective lens collects these rays and refracts them to produce the first real, inverted, and magnified image inside the microscope tube. This real image then serves as the object for the ocular lens, or eyepiece. The eyepiece further magnifies the real image and forms the final virtual image that we observe through the microscope. Because the direct transmitted light never enters the objective, the background remains black. Only the light reflected, refracted, and scattered by the specimen contributes to image formation, causing the specimen to appear bright against the dark background. This simple modification in the path of light completely changes the appearance of the image. Advantages: allows observation of living, unstained microorganisms. Provides excellent contrast for thin, transparent specimens that are difficult to visualize using bright field microscopy. Ideal for observing the motility of spirochetes and other delicate bacteria. Disadvantages: produces limited internal structural detail because the image is formed mainly by reflected and scattered light. Requires precise optical alignment, and dust or scratches can produce bright artifacts. Not suitable for thick specimens, as excessive scattering reduces image quality. Thanks for watching. Like this video and subscribe to my channel.